Let us discuss about the bryophytes now. The bryophytes are non-vascular embryophytes, which are characterized by the presence of an independent gametophytic phase and a parasitic or dependent sporophytic phase. They occur in moist and shady places. The dominant and independently existing phase is the gametophytic phase. They do not have vascular tissue. They may have rhizoids, a root-like structure for absorption of water and holding them to a solid surface. The plant body may be thallus, that is made up of undifferentiated mass of cells, or leafy or foliose. The vegetative reproduction is through fragmentation, budding or gemma. The sex organs are multicellular and they are jacketed. Two types of sex organs can be seen in them: the antheridium and archegonium. The antheridium produces antherozoids and the archegonium produces eggs or oosphere. The fertilization is within the archegonium. The zygote grows into a sporophyte, but this sporophyte grows on the body of the gametophyte. The spores are all homospores, that is, they are all of the similar types of spores. Now, in some bryophytes, we see a special stage called proteinema stage. The germination of spores produces a juvenile filamentous gametophyte, which is actually microscopic, autotrophic, and has rhizoids for attachment to the substratum. They are specialty of mosses. They give rise to the leafy proteinema or leafy stage. The bryophytes can be divided into two types: the liverworts or the hepaticopsida, or the mosses or bryopsida. Let us see the liverworts or hepaticopsida. They are the dorsiventral bryophytes with unicellular rhizoids. They occur in moist, shady places. The shape is in the form of small dorsiventral plants. 20% of them have thallus structure, whereas the 80% of them have the foliose structure. The vegetative reproduction is through gemma. Gemma are the multicellular green reproductive or asexual buds that develop in specialized structure called gemma cups. The proteinema stage is absent. The vegetative plant body is gametophyte and all members are homosporous. Example, Merchantia. Now let us discuss about the life cycle of liverworts. As I said, the liverworts, they have multicellular jacketed sex organs known as the archegonium and antheridium. Antheridium is the male sex organs and produces antherozoids. Archegonium is the female sex organ producing producing your oosphere or oogonium. Now, the antheridium is actually club-shaped and it produces biflagellated anthrazoids. And the archegonium is flask-shaped, and within the bulb of the flask, the oosphere or ovum resides. With the help of water, this biflagellated anthrazoid swims into the archegonium. Fertilization takes place within the archegonium. After this, the fertilized zygote does not undergo zygotic meiosis, but it grows into a sporophyte, which is diploid in genetic composition. This diploid sporophyte, when matures, few of the cells in this sporophyte undergo meiotic division. And this reduction or meiotic division gives rise to haploid spores. The haploid spores are liberated. They deposit or they rest on some conducive environment and start growing into a gametophyte. This gametophyte again bears antheridium and archegonium. In this way, the life cycle continues. The next are the mosses. Mosses are also called Bryopsida. They are the radially symmetrical leafy bryophytes, in which the rhizoids are multicellular. They also occur in damp, shady place and need water for fertilization. Their shape is erect and radially symmetrical. They are mainly foliose or leafy in structure. The sporophyte is differentiated into the foot, seta and capsule. As I said, a specialized proteinema stage is seen in these mosses. The proteinema stage produces lateral buds, from which the leafy stage arises. The example of liverworts is marchantia and that of mosses is funaria.